Hi, my name is Nicholas Reyes, and this is for the Local Expert Project. This topic is 3D cell culturing, scaffolds, and matrices. Now, for the first part of this presentation, it is the technique explanation. Now, 3D cell culturing is a technique that allows cells to grow in a more realistic environment in comparison to its 2D cell culturing counterpart. And when I say realistic environment, I mean more of what would resemble the inside of the human body, such as having more fluid outside of the cell and being able to have the cell move around more freely instead of on a 2D cell culturing counterpart, which is more static and in one layer. How this can be achieved is through parts called matrices and scaffolds. Matrices are what provide the three-dimensional structure for the cell growth, and examples of these are hydrogels or extracellular matrix components, ECMs. Scaffolds are components to allow the cells to adhere and grow, to allow them to organize in a 3D space. Now, which module does this specifically tie into? This technique ties into Module 5.1 specifically, as 5.1 is the introduction to cell culturing. This technique sheds light on a method in which cell culturing is not normally thought of when first thinking of cell culturing, as usually when we think of cell culturing, we think of 2D cell culturing, not 3D, and as most think of the cell culturing normally done such as 2D cell culturing. Now, what are some limitations to this specific technique? The first would be the throughput. Limitations with 3D cell culturing is that it is time consuming, which can hinder project development as it eats up too much time compared to its counterpart, 2D cell culturing. And if there are any issues along the way in 3D cell culturing, this would even set further back the project, as 3D cell culturing not only now takes more time, but in order to restart it, takes more time than 2D cell culturing. Measuring in microscopy. Since 3D cell cultures are larger in scale compared to 2D cell cultures, it can be quite difficult to measure and perform microscopy on it. If you are not familiar with 3D cell culturing and more familiar with 2D cell culturing, there would be necessary training to have to be done prior to analyzing the data that 3D cell culturing gives. Keeping the culture fed. Due to the larger size of the culture as well, it can be hard to determine where and how you will send nutrients needed for the culture to proliferate. In 2D cell culturing, when you need the culture to be fed, you can administer it to the culture pretty evenly and pretty easily. In 3D cell culturing, this can be a little bit more of a difficult task, as you need to determine where and how and for the nutrients to go. Now, what are some advantages to this technique? As indicated previously in the technique explanation, a better representation of what is happening in the body is an advantage to the technique. Since 2D cell cultures only show in 2D what is happening, it may not be the greatest indicator on what is actually happening within the body as 3D cell cultures can. 3D cell cultures show fluid flow. Compared to 2D cell culture, a 3D cell culture can include fluid flow, which is a crucial part of what is going on with cells within the environment you would naturally find them in, fluid inside the body. This can also reduce animal models. Due to 3D cell culturing would be more reliable as a method to show what is happening without the use of animal models. You'd be able to reduce the animal models overall, since a 3D model could simulate what is happening within the body. And you would not need to use any animal models, as the 3D cell culturing would be able to provide the necessary means to analyze and see what could potentially happen inside an animal. Now, what are the necessary controls you would use for this technique? What would be commonly found for the 3D cell culturing would be a negative positive control, 2D cell culture control, and a matrigel only control if you are using a matrigel. Now, going over the three figures from three different articles, the first figure is a representation of the schematics and assembly of a device that is used to perform the 3D cell culture. This is specific to the article that was in question, but the system with its components show the parts that are used, such as the gaskets, the membranes, the 3D scaffolds, top with reservoirs and microfluidic channel, the bottom with microfluidic channel, and it even gives in the other views, is in figure B, for the side views of what it would look like, and then in figure C of what it would actually look like put together, and then the culture starting with the apparatus. Now for figure 2, this figure shows a drug called gefitinib and its interaction with skin cells infected with a virus. The virus in question is in the article known as CPXV. By using a 3D cell culture method, they can see what the effects are or what would look like more so in a human body. If you were to use a 2D cell culture, you would see the effects that would be done to the skin cells on a 2D plane, but by using a 3D cells culture, you would be able to see how the cells react to the drug, but also how things flow within inside the human body and where things would go in pertaining to inside the body and how the fluid dynamics could change that. The last figure that I wanted to bring up to attention is in figure 3, which there is a lot going on here, but I want to direct attention to the A part of this figure. And all of these figures together show the regulation of the proteome in scaffold-free 3D cell cultures. The first part, which I want to shed light on more, shows the workflow of this portion of the 3D cell culture, and what is being focused on as a 3D cell culturing is the main aspect of this project. Thank you for listening, and I hope you learned a little bit more about 3D cell culturing, scaffolds, and matrices.